very much for the invitation to come to Hyderabad again. I've been a number of times and have quite a few friends here, as well as ex-colleagues and students. It's great to see the university in such good health. It's fantastic to see Hyderabad expanding with this amazing information technology and other entrepreneurial activities. And so I'm really pleased that I've come again and feel rather guilty that I've not come for the last, I think, decade or so. So I'm also honored to give this distinguished lecture. And I'm going to talk, as you can see, about modern developments in genomics, their impact on the structure and architecture of biological systems, and how we can use these very fast-moving developments in discovering new medicines, in drug discovery. But as you'll see from my talk, it's a battle. We're trying to make new medicines, new drugs, but of course the tumors in cancer and the pathogens in TB and other infectious disease fight back. So that is the broad theme of my talk. And the question is, what, as an academic, can I do to help this situation? So I'll say a little bit about recent expansion of second-generation genomics. I'll focus on cancer as one disease, but looking at it at a molecular level. I'll say quite a lot about TB, and it's not because I began to work in TB, but it's because Bill Gates sent his envoy to my company and asked whether we would use the techniques developed at great expense for cancer on TB, and he was willing to fund it. And so over the past 7 or 8 years, we've been heavily involved in TB. And one of the themes of my talk is how the two problems are so closely similar. But underlying all of this are academic studies on understanding the architecture of the kinds of targets that we might use in drug discovery, both at this large, low resolution level and in much finer detail. And I'll say quite a lot about the challenges that the genomics, these incredibly complex structures and these very detailed interactions, how they challenge our ability to cope with the big data. It's a big data problem, but one that has to be focused on individual drug candidates. And so, as I said, I will then discuss how these drug candidates, once they get onto the market and into the population, lead to resistance, and how we can understand that resistance, and some ideas at the end as to how we might fight it. So that's all about what's happening now, but I thought I would say a little bit about my history and how I reached the point that I am at the moment, because in the very early days of working with Dorothy Hodgkin, I think I learned just about everything that was important. Dorothy Hodgkin here is in her 30s, in the 1930s. Of course, I walked into the laboratory where she'd been working for the previous 30 years, 60 years later, and I walked in and found her working on these crystals. These crystals are actually crystals of insulin, and insulin, of course, is the protein hormone that's necessary to lower blood sugar levels and is used commercially for the treatment of diabetes. And I'll say a little bit about how important that is in a moment. But I was very lucky, although I'd walked into the lab 30 years after Dorothy had started the project, I was lucky to be there when we solved it. And my first impression of this molecule, it's stored in the pancreas, circulates in the blood circulation, and then finds its receptors to act upon them and change sugar levels. I first thought, this is not only very interesting, but it's also fascinatingly beautiful. And of course it was fantastic to have all of this wonderful high-profile papers on what we've been doing. And I just thought I'd show you one picture from this period of time, a little bit later, and I just want you to see how if you're a real scientist, you work on one thing from when you look like this to when you're a little bit older, and how Dorothy had the ability to focus on one theme over a long period of time. But I also want to point out how 40 years ago, about the same time as I came to India the first time, how beautiful I looked. Now, apart from all of this and the wonderful discussions you can see I was having with Dorothy Hodgkin at the time, I walked in the laboratory when she got her Nobel Prize almost the same time, so I certainly didn't contribute to it. But we had wonderful discussions, and it was amazing to be involved in this final stage in her challenge with science. But another thing struck me very strongly, and that is that, as the Vice Chancellor said, Dorothy Hodgkin had very strong political views. She was a person of the left, and in fact she was so left-wing, she was prevented from going to the United States in the McCarthy period because of her political views. However, Dorothy Hodgkin went into many companies, Novo, Wellcome, Eli Lilly. These are all large centers of capitalist activity, which made a lot of money out of making pharmaceuticals. And the general view in Oxford at the time was that probably those who weren't good enough to stay in Oxford went into companies. And so people asked me on high table in Oxford, when I became a junior research fellow, they asked me, why does Dorothy Hodgkin go into companies? Does she go to teach them? I was amazed, because I knew from visiting them that the people in Novo, Wellcome and Eli Lilly were just amazing scientists. The first reason why Dorothy went into the companies was just to talk about science. The second reason was, of course, that many of her students were there, and she had many friends, and so she was well networked in. And the third reason was that all of the molecules she studied in her life, penicillin, B12, or insulin, actually originated from industry. By interacting by industry, she stimulated her own science, and it played a major role in her Nobel Prize, penicillin and B12 in particular. And that's something I learnt and really was impressed by. So a little bit later, around 10 years later, when I had my own research group, I left Oxford and went to Sussex University. I was then working on a system. Of course, pepsin is a digestive enzyme, but this molecule renin is a relative of pepsin, which is responsible for lowering blood pressure. And I was working on this. I started working on pepsin. I realized that it was related to renin. I thought I might be able to say something about lowering blood pressure. And so the people that I'd met in this period with Dorothy Hodgkin, I went off to see in a number of other companies. I have friends there, and I asked them what we could do about this. And for 6 or 7 years, from mid-70s, I mainly worked with them in the lab, but eventually I decided we would publish some of our results, and so we published a model for this enzyme in Nature. It had a huge impact. About 30 companies wrote to me and offered me collaborative contracts, and we continued to work with quite a few. But then some amazing things happened, and these were all basic science to begin with. We were able to define the interactions of the substrate with the active site cleft, and as I looked at this very academic subject, I realized that if I could design something that fitted in these little pockets, that replaced all these hydrogen bonds, I would have a drug. And of course, most drugs at that time were made by just screening. They were what we now call phenotypic screening, and mostly we didn't know what the targets were. And so for a time, we thought about this as rational drug design. I prefer to call it structure-guided drug design, but that started in the 70s. But I was in my academic lab, and other things started happening. I looked at this molecule, and you might think, well, you can see it has two halves, that's pretty obvious, but you may not recognize that the topology of that half is identical to that half. And in fact, lots of people had looked at this after we published it for at least a year or two without realizing it. And I asked myself, why was it that these two halves were similar? Well, the argument obviously was that there was an ancestor to this protein somewhere back, and I wondered where it was. And I wondered, could I find it anywhere in any organism today? And so in those days, we did have computers and we did write computer programs, but the literature came in on paper. So I had to go to the library. I find most of my colleagues now never go to the library, they just go on the web. But I went to the library on Saturday mornings, and I looked for something like this. So this and this have the same topology, but I've made it simpler on each side, and this was the hypothesis of the ancestor. And I looked in the library on Saturday mornings for some time, and by the way, I published it, so you can see I'm not just telling you a story, you can go and look at it. In 1978, I published it again in Nature, and I looked for something. And in 1984, I found something, not where I was expecting it, but in Rous sarcoma virus. And then at that time, of course, the AIDS epidemic was growing, and we realized there was a relationship between HIV and the Rous sarcoma virus, and therefore I wondered whether in HIV there would be something looking like this. And of course, immediately all sorts of bells started ringing. And here in 84, 85, we had the sequence of HIV. It's a long single gene that codes for a polyprotein, and this polyprotein is then cleaved up into a series of genes. And the original annotation of this gene, or this genome, was in fact reverse polymerases and various other envelope proteins, but there was something missing. And what was missing was that this polyprotein had to be cut up. So was there anywhere in this polyprotein a pair of scissors? And looking at the sequence, I spotted right there the proteinase, the pair of scissors. Unfortunately, somebody else was quicker than me, and in 1985 published it in Nature before I could get it in. But what they didn't quite realize was that this molecule here must, in fact, dimerize, and it must look like renin. And therefore, we could very quickly begin to say a few things. We could say that the virus comes in here, it of course incorporates itself, it expresses a single polyprotein, and this proteinase, as a pair of scissors, cuts up the polyprotein to give you the mature virus. And before you would ever get an active and dangerous virus, this has to happen. So of course, we speculated, but my colleagues, experimentalists as we all really are, criticized me strongly, and therefore what I did was two things, was to think about the implications and try and do the experiment. Now, it took me until 1989 to determine the three-dimensional structure of this HIV proteinase, or protease, and it was precisely as we had predicted. But then there was a second thing, which I'm obviously going to have learned from Dorothy Hodgkin. Here I have a molecule, it's key to a process. Why don't I do something about making a drug which would bind that pocket? And of course, the obvious thing to do was to go to a company that was making renin antihypertensives, and I went to Pfizer, and I said to them, look, why don't you take your antihypertensive drug, and which was being studied in Groton in the US, where I would go every 2 months, and then in between I would go to Sandwich in the UK, and in Sandwich in the UK, the Pfizer labs were studying AIDS antivirals, just setting up. I said, take your drug from Groton, antihypertensive, and try it in your AIDS program. They all thought I was crazy, but of course, as you can guess, probably wouldn't be saying it otherwise, I was right. And of course, many other people, I should say, in parallel, had similar ideas. I had then two things happen to me. One is I formed a company, and the next thing that happened to me completely obliterated that. Margaret Thatcher, our Prime Minister, asked me first to advise her, and then secondly to take on an administrative role in running a research council. And I was then told by the government, although it was a good thing for scientists to have good ideas about translation of their science, in fact I was a government employee, so I should close my company or leave, which I found very surprising. But others went on, of course, to make HIV proteinase inhibitors, and this is one made in another company, Merck, and HIV positive, in fact AIDS patients, are kept alive with a cocktail of drugs, one of which is the inhibitor here. And the other thing that happened was that we could sequence this very small HIV genome and look at where the mutations were occurring in AIDS patients who were getting these drugs. And you can see that all around the HIV proteinase, and especially around the ligand here, there are mutations which have the effect of rejecting the drug, but still allowing the enzyme to function. And so that, you can see, is really a sort of blueprint for my thinking for a long time afterwards. So when I came out of my government job, I thought I would form another company, and it would be based on this model. So here we have a gene, and that is really leading to a definition of biological space, because the genes define the biological outcomes, the proteins, the gene products. These, of course, interact and define biological space. So if we were going to make drugs, surely we should use the techniques that I've been using on HIV protease, and this was over 20 years ago now. But so we should surely use this sort of thinking to choose targets. But then we should also do something else, and that is we should use this architecture to define new drugs. So this is defining chemical space, and what we have then is the task of defining biological space, identifying the targets, and defining chemical space, defining the chemicals that will be consistent with the target site. And so what we began to do is to think of how we could do that, not on an organism with just 8 or 10 gene products, but one with over 20,000. In other words, the human proteome. and as well as in pathogens. So the first thing we have to do is define the biological space, and I just want to make a few comments on that in a moment. But then we have to define the chemical space, and what happened in around 1990 is that companies started getting huge libraries. These libraries, by the way, had sometimes 1 million compounds in them, and I'll tell you a little bit about what my impression of that development was. But the other thing that was clear is that the biological space was very limited. And even 20 years later, there is no organism except for a very small bacterium where we know more than 10% of the gene products, the proteins, in terms of the architecture like this. And if we realize that many of the targets are going to be multi-protein assemblies, then the figure is almost certainly less than 2%. And even now, we probably don't know more than 85% of the functions of genes, and that's probably an overestimate on most living organisms. And so there's a lot of work to be done. And so in my academic lab, what I've done over the last 30 years really is to first begin to look at this biological space. And I just give you one example from our lab, and this is when you have a DNA double strand which breaks, both the strands break, which is a catastrophic event, and there are two ways of joining them again. One is called homologous recombination, and that means you have a sister chromatid, you have two chromatids, you can use one to provide a template for mending this, and that's called homologous recombination. I'll tell you a little bit about that in a moment, but there's another system, because in certain parts of the cell cycle you have only one copy in the cell, and then you need to use non-homologous end joining. You put a whole pile of proteins on the broken ends, and you join them up with a ligase, and that's called non-homologous end joining. And so what I've done in my lab, just to give you an idea, is here's the DNA in the center, and a few years ago these were the proteins that we knew interacted with the DNA. So we've got to define that if we would want to target it. And why would we want to target it? It's because, for example, if you have a tumor and we use radiation therapy, the radiation therapy brings in double-strand breaks. And therefore, if we wanted an effective radio treatment of tumors, we'd need to have something that stopped the cells repairing themselves. And so it's worthwhile inhibiting this system in terms of tumors. So what can we do? Well, we can get some bright people from all parts of the world. We can use small angle X-ray scattering to look at these interactions, a little low resolution. We can use electron microscopy to look at the complex structures. We can work, still not for 30 years like Dorothy did, but 5 years or even 10 years on defining these three-dimensional structures. And this is a paper that Lynn Sibanda here from Zimbabwe and Dima Chivgadze from Russia and I published together just 2 or 3 years ago. And that's a huge X-ray challenge. But we also need to know about stoichiometries, so we have to use things like nanospray mass spectrometry. And here's Qian Wu from Shanghai, from China. So let me just show you the sort of things we do. Just take these two components that are known as scaffolds, and I say what I think they do. So Qian, imagine those two molecules as dancing together. And many people, including Qian, show that if you mutate these two hands where they were holding hands, in fact, you inhibit this process. As you can imagine, they went out onto the dance floor. If there was only two there, then they're just dancing as a couple. But as more people crowd onto the floor, then what happens is you get a sort of line dance with these alternating, all experimentally defined. And then we found as you increase the concentration further, you got an even more amazing structure, perhaps not an analogy on a flat dance floor, but this is a cylinder in which these scaffold proteins form a tube. And into the end of this tube go the whole of the nucleosomes, the DNA repair equipment, and they're all brought together. And so the first point that I want to make is that if you're going to target these things, you have to define these amazing structures and really understand what the biological processes are. And one gene may equal one protein, but one functional unit involves more than one protein, usually, in a complex space and temporal, so spatial and temporal series of events. So absolutely critical to get this described. But then, of course, we can't do experiments on everything. We need to use computational methods. So how do we extend the information we've got? Well, you get somebody bright, and in this case from Germany, this is Adrian Schreier, who makes a database of everything that we know about how proteins interact. And there are 80 or more thousand protein structures now defined, and we can look at, say, a small ligand or a protein-protein interface, and we can define all the interactions, and we need to know that if we're going to understand A, how they function, and B, how we might replace them with a drug molecule. So inter and intramolecular interactions. We need to define the interactions as a number of structural interaction fingerprints. So these are mathematical concepts which are very important. We need to be able to take this molecule and divide it into fragments, because you see how this molecule interacts individually in different ways, and I'll show you why that's important in a moment. But we can store in our computer these molecules fragmented, and then, of course, we can reference to another database, and this is actually written by another one of the people who did their PhD in my lab, John Overington, who's now at the EBI. He was in my lab 25 years ago, but now he has the most amazing database of functional information. So I can ask, have you ever seen a bit of this molecule anywhere in anything that has biological activity, and particularly is it in any drug that's currently used? And so these computational techniques have to be modern computer science, and so you can see not only do I need people from different countries, I need people with different disciplines in this. And so what you can do now, this is all free on the web, you can go in, you can look at the interactions, see how they vary, and then you can take, say, the TB genome, which has 4,000 genes. We only know the functions of probably about 3/4 of those, although I've been having discussions down in Bangalore yesterday on our Sunday workshop, and I think we're doing a little bit better than that now. But we only have roughly 1/10 of these gene products where we know the architectures, and of course there's all sorts of other problems about redundancy in pathways and systems which we need to understand, which I'm not going to go. So we need to take the 10% of structures we know, and then we need to look at the other gene products and work out what the architecture looks like. And the way we've done this is to use a series of software that I've developed in the lab, and we tend to use our own, but other people have, and I should say since, because we were the first ones ever to code these up in the 1980s, what we call knowledge-based prediction. So what we take, for example, is a protein, and we divide it into protein fragments, and we learn how in evolution we might assemble another one of the homologous family. And this is called homology modeling or comparative modeling. The first program we wrote is actually being used commercially for around 20 years after this date. The second one we wrote, which is Modeler, written by Andre Shally. He's now a professor in UCSF in California. That has already gone over 7,000 citations, or just about that. It's a little bit out of date, this. And you can see that other programs have been rather well cited on the whole, and some of the recent ones, I haven't put that citation down, it's still, I think, well over 100, but obviously all of them are contributing to trying to build models of the rest of the proteome. And I'd just like to point out, composer, fugue, and rapper are all musical terms, and that's because I spent my youth as a semi-professional jazz musician and would have much rather done that than science, but it was wonderful to find how sequences and structures were as musical as some of the music that I wanted to play. I never could, I wasn't good enough, but I could reproduce it here. By the way, this guy insisted on calling the program Modeler, but he married an opera singer in the end, so I forgave him. And what we can do from that is to build up a database. So let's take the TB database, which we call Chopin, musical term. Chopin died of TB, as you know, and we can build models of that. Around 70% are either likely or pretty likely models. Quite a lot of the other models out here are less likely, and some we just can't do anything about. But what we're doing here is using evolution to inform from one family to another family in the same genome, and from one organism to another organism, and build models. Of course, they're not that accurate, but they are useful. Here's an example. This is QCRB. It's a cytochrome BC complex, and my collaborators on the TB program suddenly emailed me to say they had a molecule, and they're at NIH, Cliff Barry, said they had a molecule that hit this system. Could I build a model of it, and where did it go? Well, we could do it pretty easily. Here it was. It has 3 components, and we hypothesized, because we knew from other homologs that ligands were likely to bind in there. And then they sent us some further information, and that was that there were 2 mutations right here which were affecting this molecule in terms of drug resistance. So suddenly we had a model, and there we had a check on it. At least it passed the test, because it doesn't mean it's absolutely right, but you can use these models and computing to inform the process of understanding drug targets. And then, of course, we want to explore chemical space. And I better speed up a little bit. Of course, what happened in the 1990s is that the chemical libraries grew larger and larger. Companies went and looked at their own libraries. Combinatorial chemistry was said to be the answer. It clearly wasn't. It didn't explore chemical space in a very effective way. Maybe some of you will argue with me on that. And they searched the world for natural compounds. And the general rules, which are called the rule of 5, are that they had to be molecular weight 500, and the library sizes needed to be over 100K. And there was great optimism in the early 90s that we were going somewhere. But here's what happened. For every 8,000 compounds, not in the library, but modifications of them made, only 8 get into clinical trials. And roughly 1 of those on the average was getting onto the market. This is, in fact, 13, 14 years ago when these statistics became evident. And the figure at those dates was about $1 billion to a drug. It's now around 2 billion. And the reason it's so expensive is because so many don't make it into the market, and you have to factor those into the costs. So you've got a huge cost. And here's what happened. You can see between 1980 and around 2000, exponential increase of research and development investment, but the number of new drug approvals didn't increase. All that effort led to no increase of productivity. And so many of us, and me included, released from the restraints of my government job, started to think how we would explore chemical space. And this is one of the ideas that people came up with, the one that we did. Other people had similar ideas. And that is, supposing we had smaller molecules, not 500 molecular weight, but 300, then the number of different molecules you could have would be much smaller. So to explore chemical space with very small fragments is going to involve a smaller library. But what's the problem? Well, the problem is they won't be biologically active, and they probably, certainly not on a cell, they may have some biological activity against a target. So you need to use other techniques, some physical techniques. So what we proposed was that if we knew the active site of a drug target, and we could imagine that there were 3 sites, and we had a library of 1,000, we could explore those 1,000 molecules to see whether they bound there, there, and there with the same library. But we'd have to detect them with biophysical methods or X-ray methods to see where they went. And I thought in 1997-8, what we would do would be to cross-link them all. In fact, it turned out that the best way was to grow them. And so then what happened is we proposed to venture capital that we would have an experimental system, we'd have known structures, and we would screen those with a very small library using high throughput biophysical and X-ray methods. And sooner or later we will get one that binds, although I sit here sometimes wondering whether I will, and there you are. That's a fragment bound. You wouldn't get it in a biological activity, but you can spot it and define it using the sort of methods, X-ray methods, NMR, and many other methods. And then we get chemists in to elaborate it. And so that's the general idea. And then I went to 2 students of mine. Well, I should say this one, Harun Jyoti, was really, as much as anybody, the driver of this. And he was in Glaxo Wellcome at the time. Here's Chris Abel, who's another academic, professor of chemistry, and the 3 of us got together and eventually persuaded yet another one of my students to give us $500,000. And what we did is we had 2 people in my lab and 1 in this guy's lab, and we tested out the idea. We could see these small fragments and then begin to do something with them. And within 6 months, and totally finished within a year, we showed that we could. We then advertised ourselves. We raised $20 million, and then we recreated our academic lab, the chemistry lab and the biology lab, up in the science park, a couple of miles away from my academic lab. And then we built a big building, of course, and we had all sorts of collaborations, but they were all based on getting a target. We'd have around 30 chemists, by the way, and we make the chemists sit round the computer and say, well, look, this fragment binds. You can see where it is. Do you like this one? Can you chemically modify this one? Or shall we try another one? Shall we modify it here? If so, what should we put? Or should we modify it along this vector? And as we did that, we showed nearly 50 times now that we could get from a millimolar molecule down to a nanomolar molecule within about 6 months. I was pretty surprised that it was so effective. And once we're down at nanomolar, then we could go into the clinical stage, and we had lots of clinical collaborators by this time, and do a mouse xenograph. That's where you put a piece of the tumor on the mouse. It usually grows like this, but if you add your compound, you hope that it's going to grow much more slowly. And in many cases we found it did, and then we could go into clinical trials. And after 12, 14 years, we had 8 drugs in clinical trials, and we were running out of money. And so we made 2 deals. One was to allow us to continue the trials, still calling the company Aztecs, but by having a lot of investment elsewhere. And then 3 months ago, roughly 2-3 months ago, we finally sold it. And this is Harun. I'm totally dependent, by the way, on Indian students in my lab. Not all of them have stayed in India, or come back to India. Harun was born in the Punjab and has stayed in England. But you can see it was bought for $886 million, so it wasn't a bad idea after all. But then, of course, I'm an academic, so what should I be doing now? That's the important thing. The other important thing is that the 100 people, many of them my students, still have a job, and we're re-recruiting now over 100 scientists, and we still have a company called Aztecs on the Science Park. But what are the challenges for academia? Well, private capital and large pharma have been pretty unsuccessful in doing anything with tuberculosis, malaria, and many other infectious diseases. And then, of course, also, on the whole, we've targeted single proteins like kinases or proteases, and these have led to, in my view, a lack of selectivity. So we've had 2 academic programs. We spun it back from the company into our academic lab and started working on TB and on cancer. So, of course, the TB problem used to be a major problem in England, where 200 years ago, 1 in 4 deaths were caused by TB. My grandfather died of TB. I was told that nobody would ever die again of it. But of course, even in the UK, it's a growing problem. And in many parts of Africa, it's a major problem. And that's because we had a lot of first-line drugs 30, 40 years ago. The vaccine is not terribly effective. There's a long therapy, it's several months, it's complicated, it leads to non-compliance, and you've got resistance rising. And there is actually now 1 drug just in the last year, but almost no activity from companies. And you can see that it affects Southern Africa, and my wife comes from that area, so it's a very personal thing. And there's quite a lot of problem in India, Russia, and in many other parts of the world. Bill Gates, I had dinner with Melinda Gates once when she told me that it was really her idea that they would work on TB, because she was watching a film on the telly, and she went and asked Bill, did he agree that it was a real problem and they should do something about it? And she said, well, what Bill did is went and looked in the web for a number of hours and came back and said, we agree. And as you know, they've invested with 1 or 2 other people 20 billion. And some of that money has come into my lab. This is why it's so important to me. This is my wife, who's quite a famous research scientist. These are my daughters. One's a lawyer, learning quite a lot of money in the city now, and the other's a qualified medical doctor. This was 2 or 3 years ago. But these are my grandnephews, and our relatives in Africa. We've had 1 person die of AIDS and 2 people not die, but have had tuberculosis. It's a beautiful country, but it has a real problem, so it's a personal problem. So here's the frontline drugs, and here's the second line. What we decided to do in the lab was to look at one of these and see whether we can enhance it. This is ethionamide, which is activated by an enzyme ETHA, and this is controlled by a transcriptional repressor, ETHR. And if we block this, we would then increase the efficacy of this known drug. I'm going through this very quickly, and I can talk about the biology if you want, but I just want to make a point. So here's the structure, not done originally in our lab, but we can take the structure and we can then show that what, in fact, a ligand binding here does is to change the distance between these 2 globular regions and change the ability to bind to DNA. And so what we did is to set up, and we've done this on TB targets in the academic lab around 20 times. We have an initial screen where we screen using the 1,000 fragments, just as we do in the company. Here's my research team. Just to make the point, it's led by an Indian who has now got UK citizenship, although he had an offer back in India. I was rather sad he didn't come back. And we have an Indian student working on it. We also have a Portuguese and a Polish. What we've done is to set up thermal shift assays, surface plasma resonance assays, so we can watch the fragments binding. And then we've done structure, thermodynamics and dynamics, and we can see lots of fragments. This is 5 fragment hits superposed. And I think you can just put this in front of a good chemist and they'll make you a molecule out of that mixture. And you can get this just in a few days. It's just amazingly quick how you can get these fragment structures. And what we've done with all of this is then to take these molecules and go around in cycles elaborating the structures. And you can see again we're working between physicists, chemists, computer scientists. Here's the chemist Chris Abel, my collaborator. So then very quickly we can do the same thing on TB. I'm running a little bit out of time, but there's a real problem in these, we can do the same thing on cancer. These multi-component systems are incredibly important in these areas, but the problem is that if you take a classical target, like the target of Gleevec, which is an anti-cancer agent that hits the tyrosine kinase, here's the Gleevec and here's the kinase. I put a coat on it, you can see it's completely buried, it's ligand deficient. If I take a protein-protein interaction, like an assembly, I take this rather flexible peptide away from here, you can see you have very, very small pockets. They're very difficult to target. That's why we had to do something in academia. And here's what we decided to do. I've been collaborating for 20 years on the MET receptor, which is the target of the hepatocyte growth factor. And when the hepatocyte growth factor hits cells, they scatter, they all move around. This is critical in tissue development, but of course, in cancer, it might lead to metastasis. 40% of metastasis are caused by mutations and problems with the MET receptor. I just want to show you very quickly, we can define the structure, we have detailed pictures, several years' work here of the whole receptor system, collaborations between physicists and a medic here, Emana, who went ahead. We have a German scientist here, and you can see this is a surface plasma resonance hit. This is just a little academic program, really. We can do structures of these ligands on one of the components of the complex, and then we can use an alpha screen to see whether we're getting indications of a hit. So this is all fairly recent work, but that's just an academic screen on something that most people thought was not going to be druggable. And so the point that I'm making here is you need a multidisciplinary team, but you can do these things in academia and do them quite effectively. Here's another example, which is the other part of double-strand break repair. And here the Indian component was the rather famous, actually medically trained. He's in the hospital. This is Ashok Venkataraman in Cambridge, who's head of the Hutch MRC Cancer Centre, and he's got a unit in NCBS in Bangalore as well. And this is a paper we published in Nature of the interaction between the ATPase or the recombinase and the BRCA2. And what we noticed was that there were little pockets in here, and so as we were writing this up for Nature, we took out patents. And I've taken out patents on lots of things that I put into Nature. They're not incompatible. So everything I'm telling you about comes out of our academic lab. All of this was actually done in my lab, although it was suggested by Ashok. And what we can then do is we can look at the target, we can look at the pockets, we can see how they're important are by looking at the sequence variation. We can look and identify these pockets, realize they're very, very small, but we can then use our methods to target them. We need assays. I'm not going to talk about this, but we set these up with fluorescent labels. And then actually we sent this out to peer review in academia and industry, and everybody said it's impossible. We have done 740 crystal structures, and I have 80 hits of detail, so referees get things wrong quite often. And over the time, we've now taken this target from millimolar. It's taken a bit longer than the ones in the company because it's a more difficult target, down to nanomolar, and we're now going to take it through to preclinical. So that's what we're doing at the moment. Now, just to finish, I wanted to comment that there's a similarity between cancer, where the new second-generation sequencing is defining all the positions of mutations which occur in the somatic mutations in individual tumors. We, of course, have the example of HIV protease, and we can do the same thing in TB. And so part of my lab, the computational part, has now been thinking again about methods that we could do to solve this. Now, of course, we've known quite a lot about single mutations in Mendelian inherited diseases. Somatic mutations are complex diseases, very much more difficult to identify, and to identify which are drivers and which are passengers. This is another problem we're looking at. It's a Mendelian cancer problem, again led by an Indian. Just to make the point repeatedly, absolutely central to my lab, so please do keep coming. This is a paper we published originally, as you can see, in 1997 with Srinivasan, who's at Bangalore now. He's a professor and a member of the academy. This is a program where we simulated a thermodynamic cycle using knowledge and probabilities, actually, of substitutions. Here I've got a computer scientist who arrived and had a crazy idea, and I thought, well, it's not really worth trying, but I better let him do it. And this is using graph-based signatures, which I haven't time to describe, to see the effects of mutations. And it turns out these two are complementary, and what we can do is to go through, this is a whole series of kinase domains from the somatic mutations, the cosmic database, but we can also do that in TB, where the mutations are becoming a major problem, and we can begin to understand them. And I guess the question we have now is whether we can make drugs to those mutations that occur, say, 40-50% of the time. And that's a challenge for the next generation. So just in finishing, you can see that problems arise, and it's a battle. We make a drug, and the bug fights back. We need to keep doing our basic science. So all of these things that I've talked about ended up with papers in pretty good journals. Just to make the point, this is Nature, not quite so good, the MTB, but PNAS. So you can publish stuff in really basic, high-profile journals, and at the same time, you can form companies that can make a lot of money and make real drugs, and you can keep interacting on this. You don't need to do one or the other. And then you see there are two other things you need. You need a very mixed population. Woman, 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 woman. Not quite as many as I'd like in the group, but having worked with Dorothy Hodgkin, I can see their power. I have to say, I went to the meeting in Mysore, and there wasn't a single woman on the program speaking. I think this is a challenge you have in India. The second problem is to get mathematicians, physicists, engineers, computer scientists, chemists, biochemists, and medics all talking to each other. And our structure, both in industry and in academia, doesn't really help that. And that's a challenge. We've got to get people working in groups. And then finally, I've stressed the different nationalities. I think that the different cultural differences helps in getting much more imaginative hypotheses in moving science forward. We can do, or could do, when it's a little larger, 37 languages in my group, and I'm proud to say I have two English people here, and that's probably a rather larger number than it sometimes. So it's a multinational, multidisciplinary, multi-gender project. And I hope then that what I've done is to really describe what a lot of fun I've had out of the science, how beautiful, how exciting it is, but also try to describe how it can be useful, but in being useful, you can still do your basic science. And that's my message. Thank you very much.